In this module, we'll talk about how there's more to you than your genetic code. We'll explore the balance between nature and nurture that influences our genetics. Except where otherwise indicated, this content is available for use under CC BY 40. In 2010, many people learned about the idea that our DNA does not dictate our future. In particular, the idea of epigenetics was introduced to the world, outside of scientists in the field of genomics and epigenomics. In this module, we will begin to explore why your DNA is not your destiny, as highlighted in the 2010 Times cover page. In particular, we will explore the concept of epigenetics and begin to understand why the choices we make voluntarily and involuntarily can affect our genes and health outcomes. In plain terms, we are considering the differences between nature and nurture. Should we be more concerned about the DNA we inherit from our biological parents? Alternatively, should we be more concerned about our environment? Some people think the balance is tilted, where our DNA dictates everything about our health. They might think our experiences have very little effect on our health. However, in most recent years it has become clear that our experiences are equally as important to our health. There isn't a right answer to which perspective is correct. Our goal is for you to appreciate the existence of a nature and nurture balance system. This system influences how our external experiences and inherited DNA influence our health. The three balances represent different stages of a nature-nurture spectrum. In reality, the balance can be anywhere on this spectrum depending on what traits we are examining. Before we proceed to our lesson, let's use this opportunity to outline the topics covered. First, we will review some essentials in genetics and genomics. We will cover concepts on the central dogma and genome packaging to familiarize you with their role in nature. We will then explore in more detail the role of histone tails in DNA packaging as well as the epigenome. It's important we understand and distinguish between the role of histone tails and our physical DNA sequence. The ability to distinguish between our genes and DNA packaging will be key to how we distinguish between the genome and epigenome. We will then discuss the importance of our external and internal exposure and its role in the nature and nurture balance. In particular, we will introduce the concept of the exposome. Finally, we will end this lesson with a brief overview of how our understanding of exposure, genetics and epigenetics will help guide biomedical breakthroughs and get us closer to achieving personalized medicine. The field of genetics explores how we inherit traits from our parents and how these traits shape many aspects of our physical characteristics, how our cells work, and even our susceptibility to certain illnesses. Our genes, made up of DNA, get turned into our RNA and then proteins. It's these RNA and protein molecules that control how genes work and regulate various functions in organisms. This process is also known as the central dogma. The central dogma explains how changes in DNA affect the creation of our RNA and proteins, which in turn control biological functions. Essentially, alterations in DNA can determine observable traits such as hair or eye color, as well as the likelihood of certain diseases. DNA is packaged tightly into chromosomes and can be visualized directly with a powerful microscope. In total, we have 22 pairs of autosomal chromosomes and 1 pair of sex chromosomes that code for all cellular function, which begins at fertilization. The collection of all 23 chromosome pairs makes up our genome, which can be found in all cells. For example, the genome can be found in epithelial cells collected in a cheek swab. Understanding how DNA shapes the identity and function of cells hinges on how it's packed into chromosomes within a cell. Chromosomes consist of specially condensed DNA called chromatin. Every cell in your body has a complete set of genetic instructions stored in its nucleus. However, not all genes are active in every cell all the time. The actual structure of a chromosome is complex and helps determine genes that should be actively turned on or transcribed at any given point in time. At times there can be a region of the chromosome packed to accommodate open chromatin, which indicates a region of the genome that should be transcribed. Similarly, there are regions of the chromosome that are tightly packed to form condensed chromatin. When chromatin is condensed, transcription can't occur. There are many open and closed chromatin regions in a chromosome. Chromatin patterns in one cell might look very different from those in another cell. Turning all of these genes on or off through transcription is what makes observable traits such as hair or eye color or susceptibility to diseases. Now, let's talk about how packaging DNA works. Histones are a type of protein responsible for packaging DNA into chromosomes. There are 5 classes of histone proteins, and each class of these proteins contain a histone tail that is rich in lysine and arginine. These amino acids aid in DNA packaging. Histones are proteins made during translation. Translation uses RNA as a template that contains the identity of amino acids needed to make proteins. In total, there are 20 amino acids that are uniquely arranged to make proteins. These amino acids have unique chemical and biochemical properties. The identity of these amino acids and their characteristics are dictated by side chains. For example, amino acids can be positively or negatively charged. As highlighted earlier, histones made during translation have histone tails rich in lysine and arginine. Lysines and arginines are positively charged amino acids. The lysines and arginines found in histones and along the histone tail effectively makes histone proteins positively charged. DNA, however, is negatively charged. This is the result of the phosphate group found in nucleotides. As you remember from chemistry, positive and negative charges attract each other. Therefore, the histone tails aid binding of DNA needed for DNA packaging through protein nucleic acid interactions. Histones come together in groups of 8. This grouping consists of 2 copies of each class of histone. When DNA wraps around this discrete set of 8 units, the histone tails are exposed, and the overall structure formed is called the nucleosome core. The histone tails pick up a variety of modifications. These regulate how tightly packed DNA will be. In addition, this packing of DNA regulates whether DNA will be transcribed to RNA. For example, acetylation modifications turn transcription on. In other words, they activate the genes due to loose DNA packaging that leads to open chromatin. However, methylation modifications on the histone tail can tightly pack DNA that leads to condensed chromatin and turns off transcription. How does what we know about DNA and open and condensed chromatin tie into nature versus nurture? The histone modifications like methylation control whether chromatin will be condensed or open. This regulation on chromatin is different from genomic and genetic regulation. Regulation that affects chromatin folding is epigenetic and epigenomic, as it is independent to the genes inherited from parents. Regulation of chromatin works differently. You can think of it as having an extra layer to how we study and think about health outcomes. Unlike genetic and genomic regulation, our direct and indirect lifestyle choices can change epigenetic and epigenomic regulation. What remains is how we categorize the risk associated with exposure to certain lifestyles. Thus, the choices that we make or are made for us have the potential to influence the epigenome, and these epigenetic changes can become a major influencer to one's health. Let's return to our nature versus nurture debate. You can think of nature as genetic and genomic regulation, and nurture as epigenetic and epigenomic regulation. We are made up of a balance of the 2 forces. Let's talk now about some of the forces that affect our epigenome, collectively known as the exposome. Truthfully, our lifestyle choices are not the only exposure we should be concerned about. Exposures that alter our health can be the result of our ecosystem, social and socio-economic status, as well as physical-chemical interactions. Our lifetime exposures to external factors broadly make up our exposome. One's exposome evolves over the course of their lifetime. This evolution is guided by external environmental factors that reflect direct and indirect exposure choices. These environmental factors influence our internal environment. The changes to our internal environments alter the nature and nurture balance that, in turn, influences gene expression. The changes made to gene expression can potentially put people at risk for bad health outcomes. Thus, linking exposure to the nature-nurture balance is key to determining health outcomes. We know the environment can affect us at the molecular level. What can we do about it? Fortunately, the advances in scientific tools and research have paved the way for studying the exposome, the epigenome, and also the genome. These advances allow us to change the ways we approach medicine and how we treat different diseases. We currently utilize a one-size-fits-all approach to treating different diseases, despite the global diversity that exists in our exposure. Even when two genomes are quite similar, the impact of exposure on our genome and epigenome can result in outcomes that may look similar on the surface, but are mechanistically very different. Advancements in science and scientific research allow us to consider the role of the exposome, the genome, and the epigenome in treating people. This scientific approach allows us to break away from a one-size-fits-all approach to medicine and brings us closer to understanding the unique health needs of people. When we consider the individual health needs of people, we are able to design therapeutics that addresses their unique health needs. This future approach to medicine is called precision medicine. In precision medicine, a person's genes and exposure is used to precisely identify the appropriate therapy to meet their needs. Let's review what we have learned. We learned that the central dogma is influenced by genome packaging into chromatin. Histones and histone modifications are important for determining how chromatin is condensed. External and internal exposures are known as the exposome and can affect which genes are active. Finally, better understanding both the genome and epigenome, otherwise known as nature and nurture, can accelerate breakthroughs in achieving personalized medicine. Thank you to our image and icon sources. Thank you to everyone who contributed.